The title of today's webinar is Advanced 3D Cell Models for Cancer Biology Using Real-Time Live Cell Analysis. And in this webinar, I hope to demonstrate how this approach of live cell monitoring and real-time quantification can provide further biological awareness, which may be missed possibly with more traditional endpoint approaches. I will briefly describe validated protocols for creating two types of 3D multicellular tumor spheroid models, and how biological parameters such as the growth and viability can be returned through the capture and analysis of bright field images using integrated software designed specifically for 3D cultures, and finally demonstrate how the development of more complex co-culture models can be investigated using the IncuCyte S3. I think you would agree there's an ever-increasing drive to improve the rate of success of anti-cancer treatments transitioning to the clinic. An accepted or very much acknowledged way to achieve this is to more closely align in vitro cellular models with each stage of preclinical, animal and patient models. The example on the right-hand side of this graph describes some ways to achieve that. So, for example, better access and use to patient-specific material can help, as well as better understanding of heterogeneity and the importance of the tumor microenvironment. In addition, advances in gene manipulation techniques, as well as improved techniques for tissue renewal, can aid further. Incorporating 3D models is very much accepted, if not mandated, in achieving more physiologically relevant in vitro cancer models. Here I want to spend a little time briefly describing two 3D models of multicellular tumor spheroids and perhaps why you would choose one over the other. Firstly, methods that produce uniform and very much size-controlled 3D spheroids, such as single spheroids, allow you not only to recreate the spatial distribution, or rather zonal regions of proliferating quiescent and necrotic cells, which are very much characteristic of solid tumours, but also the physiochemical environment with respect to nutrient waste, oxygen, and nutrient gradients. This exquisite standardization of size and the formation of a single spheroid in each well of a microtiter plate makes this approach very compatible with industrial cancer drug discovery programs and achieving very strict high-throughput screening performance metrics. Conversely, the lack of standard size and morphology is very much characteristic of multi-tumor spheroid models. Here, spheroids are much smaller. They form and grow by natural aggregation on or in an extracellular matrix that closely mimics the tumor microenvironment. This model lends itself very nicely to capturing and monitoring biology that arises from naturally occurring heterogeneous spheroids and is well suited to use with organoids and patient-specific material. Now, you may be very familiar with the several published approaches to generating single spheroids, but here I will just focus on the formation of single spheroids in ULA round-bottom plates. The schematic shown here describes a validated protocol for the formation and treatment of single spheroids in not only 96-well plates, but 384-well plates. Again, there are several ways to create multispheroids using ECMs. This schematic describes a workflow for creating and monitoring multispheroids which have been created on a base of Matrigel in 96-well flat-bottom plates. Although not shown here, we also describe a protocol for tumor multispheroids created within a sandwich, if you will, of Matrigel. That is to say, cells are seeded on a layer of Matrigel followed by an additional layer of matrigel which is added on top. Both of these protocols described have been validated for use on the IncuCyte S3, and coupled with new integrated software specifically designed for these 3D culture models, allows for the continuous life cell monitoring and quantification of biological parameters such as spheroid morphology, growth, and viability. So here are some videos of single and multi-tumor spheroids creating the protocols I've just described. You can see that the IncuCyte captures both high definition phase contrast as well as bright field images. Some of you may already be very familiar with the challenges of consistently segmenting or masking phase objects. In particular, phase images of very large single spheroids of greater than 500 microns in diameter, where the center of the spheroid will often appear out of focus. And equally, poor phase contrast images of multi-spheroids that sit on the meniscus of an extracellular gel. Here, we use a proprietary method to capture depth of focus bright field images from both of these models. Note how the IncuCyte is able to consistently mask these bright field images over time, as shown by the yellow outline mask on the bright field images. So this slide illustrates how the size, or rather the area of a population of multispheroids can be tracked and quantified over time using a label-free approach, that is, using the bright field images displayed here. You can see that the S3 is able to achieve this for multispheroids which have distinctly different morphologies. So on the top panel, this is illustrated with a round morphology characteristic of MCF7 spheroids, and on the bottom panel, a more branched or stellate morphology of the MDA cells, which is characteristic of an invasive phenotype. The graphs on the right-hand side show the different temporal profiles of the two cell types, tracking not only the growth, but the lack of growth or death induced by the treatment of a known cytotoxic agent, camptothecin. I just want to introduce you to a new feature that's specific to the S3 and not available on previous versions of the IncuCyte. This is the DeepView. You can see it's a simultaneous visualization of all images in each well of your microtiter plate, enabling very rapid identification and assessment of wells. There's a zoom-in function that allows you to interrogate a region of interest, and the visual is continually refreshed as you scroll through time. Here you can see a matrix set up assessing the growth and shrinkage of 4 tumor multispheroids at 2 densities. Here are the phase images from all 96 wells, the bright field images, as well as the bright field overlay mask for each object within the well, and quantification of those images in the form of a plate view showing multispheroid growth across all treatment groups over a 10-day period can be readily visualized. Again, this plate view is updated in real time, where images are automatically analyzed as soon as they are captured. So returning to how assessment of images and videos through time can provide additional biological insight. Here is some validation data showing how the size of A549 multispheroids is dependent on cell seeding density. So hopefully you can see on the left panel, cells seeded at the higher density achieve a growth plateau sooner than cells seeded at the lower density of 1,000 cells per well. Something you may well predict and reasonably speculate is due to a depletion of nutrients over the 10 days in culture. The middle plot quantifies the number of multispheroids that form for each seeding density and how that changes over time. Interestingly, this decreases for the highest seeding density, the dark blue line, 4,000 cells per well, but remains, I would say, relatively constant for the lowest seeding density. Closer inspection of the videos and images from this experiment show that this is due to neighbouring multispheroids merging to form a single larger spheroid. Finally, looking at the average size metric, so that's the panel on the right-hand side, you can see that cells seeded at all three densities have the same growth rates over the first 72 hours post-formation. And cells seeded at the higher density have the smallest average size. And I guess since they are further apart from each other, the lower density multispheroids are less likely to merge and have more room to continue to grow to a larger average size. I think you'd agree that this type of matrix or validation experiment described, with these associated metrics, allows you to easily define final assay concentrations accordingly. For example, knowing what seeding density to use if you wanted to minimize the impact of merging in your experiment. So earlier, I showed you how bright field image capture from control wells and treated wells with a known cytotoxic agent could be used to quantify growth and death of multispheroids. Now, one might argue that this is not actually death, but possibly the lack of growth we are tracking in treated wells. And a way to verify this would be to use a cell health reagent such as IncuCyte Cytotox Green. This is a non-perturbing dye to normal cells, which has a very low intrinsic background fluorescence, and only becomes fluorescent upon binding to DNA in unhealthy cells. The bright field videos on the top panel show the control spheroid continues to grow while the treated spheroid does not. And this can be quantified and displayed in the top right-hand plot tracking spheroid area. The fluorescence videos on the lower panel show firstly a strong fluorescent signal in the treated, much smaller spheroid, and secondly, a much dimmer signal in the larger control spheroid. And this fluorescence is revealed much later compared to the dying well. Quantification of this cytotoxic mode of death can now be determined without the need to perform fluorescent object masking, which can be very subjective, and the interpretation of results often influenced by thresholds the user might set for fluorescent segmentation. Now, a new metric within the S3 specific to single spheroids and multispheroid protocols is described, and this returns the intensity of the fluorescent signal within the bright field boundary, i.e. very much with respect to the size of the spheroid. And you can see that this is verified by looking at the bottom right-hand plot, which shows an increase in the intensity of the green signal in the treated wells. Now, the use of the IncuCyte cell health reagents can be extended for use with multispheroids. Here we have an example of MDA MB231 multispheroids treated with camptothecin, just like the previous example. And as with single spheroids, quantification of bright field images in the top panel illustrates the continued growth in the control well, so that's in blue, and a lack or minimal growth in grey of the treated multispheroid. The use of annexin 5 here in this example and the quantification of the green fluorescence intensity in these images illustrates apoptotic death by camptothecin. Now, these dual readouts can be further multiplexed with an alternative measure of spheroid growth and viability. So here, these cells stably express the red fluorescent protein, and they're being referred to as MDA nuclide red cells. You can see the increase in the intensity of the red fluorescent signal in the growing spheroids and the decline observed in the treated spheroids, shown in the bottom panel of videos. The corresponding time course demonstrates how the fluorescent signal from stably integrated FPs can be used as a potential surrogate for monitoring growth and viability. So of course everything I've shown you for monitoring and quantifying growth of spheroids in a label-free manner can easily be achieved using fluorescence as I've just described. Here we have another example of a DeepView illustration, this time of MCF7 multispheroids stably expressing RFP. And you can see we can rapidly determine the impact on the potency of 3 cytotoxic agents on multispheroid growth and viability. And finally, the plate view shows the quantification of both those bright field images and fluorescence intensity signal across the treatment groups. So here, from a single well, we're able to discern both the label-free metrics from the bright field images as well as the fluorescent viability metrics from the fluorescent signal. So changing tack slightly, I think when I started, I tried to highlight that advanced 3D spheroid models are becoming increasingly important and being accepted as a key tool to enable better assessment of cancer, and in this instance, cancer immunotherapy. Here is an example of a 3D single spheroid tumour model looking at the growth and immune destruction of a tumour spheroid. The videos on the left show how the red fluorescent protein expressed in these SCOV spheroids continues to increase in intensity through time as the spheroid grows. The middle video shows that there is minimal death, i.e. minimal loss of fluorescent signal occurring in this spheroid upon inclusion of non-activated PBMCs. Interesting to note how the PBMCs initially surround the spheroid but then appear to retract and continue to move somewhat randomly around the spheroid. In stark contrast, accepting activated PBMCs display enhanced and targeted killing of the spheroid, and this is manifested as a loss in fluorescent signal and lack of growth of the spheroid. So all these phases of immune-targeted death that I've just described can be easily quantified on the IncuCyte S3. We observe a concentration-dependent inhibition of SCOV spheroid growth by Herceptin, so that's through the loss of the fluorescent signal from the dying spheroid. And we can demonstrate that this is a specific effect since we don't see the same observation in HER2 negative spheroids A549. We've also gone on to show how there is an activator dependence on tumour spheroid cytotoxicity, where kinetics of death between different subpopulations of immune cells occurs. So, for example, we've observed that T-cell mediated death via the activation with anti-CD3 and IL-2 occurs more rapidly than NK mediated death with a different range of activators. But it's also worth reminding ourselves that this may reflect potential differences in the concentrations of subpopulations of these immune cells in the PBMC prep, and also take into account potential donor-to-donor variations. So again, with this increasing drive to build more translational 3D cancer models, recapitulating the tumour microenvironment is key to achieving this. So here I wanted to demonstrate how we could monitor the effects of stromal cells, and in this case, normal human dermal fibroblasts, on tumour multispheroid morphology. So you can see, just by focusing on the videos on the left-hand side here, the inclusion of these fibroblasts with MDA multispheroids encourages them to form a more stellate morphology very early on in the time course of this experiment. But as that experiment progresses, you can see that both cultures take on a very much equivalent morphology. Interestingly, it's only in the presence of fibroblasts that SKBR3 cells form more compact spheroids. So you're now looking at the right-hand panel of videos and homing in on the bottom one. And in the absence of these fibroblasts, I think you'd agree that they remain very much as loose aggregates. So here is a quantification of the SKBR3 videos that you've just seen on the previous slide, where the total area metric indeed aligns with what you've just seen, in that there is the greatest area coverage with SKBR3s in the absence of fibroblasts. These two cultures have been treated with camptothecin, that cytotoxic agent I spoke about earlier, and an area under curve analysis performed of the whole time course to show that camptothecin displays the same potency across both treatment groups. Taking this one step further and performing more detailed compound profiling, here we illustrate differential pharmacology, in this example, across 4 tumour breast cell lines. So, as alluded to earlier, the broad-spectrum anti-cancer compound camptothecin, as you would probably predict, is showing equipotency across all 4 cell types. In contrast, lapitinib, which is a dual HER1, HER2 tyrosine kinase inhibitor, shows marked activity at both SKBR3 and BT474 multispheroids, with marginal inhibition displayed in the other 2 cell types. In contrast, the progesterone receptor antagonist ZK164015 strongly inhibits the growth of MCF7 multispheroids, and then followed by SKBR3s and BT474s. Interestingly to note, the rank order of potency of lapitinib and the ZK compound align very nicely with the known expression profile of the receptors that these compounds are known to target. Now, having demonstrated the S3's ability to acquire and analyze images from 3D multispheroid co-culture models, you may consider using fluorescently labeled endothelial cells instead of fibroblasts, or possibly studying the impact of immune cells and stromal cells on tumour spheroids in a triculture setting. So in summary, I hope I've demonstrated to you the key features of the new IncuCyte S3 software, which is, as I've said, has been designed specifically for monitoring 3D cultures. Given you a flavour of the integrated workflows and the automated life cell analysis for both single and multi-spheroids, and how real-time image-based approaches enable assessment of spheroid morphology, how spheroid growth and viability can be determined in a label-free manner and fluorescence way by multiplexing either with IncuCyte cell health reagents or using stably integrated fluorescent proteins, and how the complexity of both single and multispheroid monocultures can be increased through the addition of immune cells or stromal cells, and the study of their effects can be performed on the IncuCyte. So if you're interested in any additional information on the areas I've touched on today, whether that be the software, the protocols, or indeed the use of these cell health reagents described, they can be found by following the links on this slide.